in GLP part. So my name is Amol Naragude, and we'll see the one by one this point. So first of all, why the quality matters in a pharma? Because it related to the medicine directly to the health, and any mistake can risk the lives, and hence it regulation of the by the GMP and GDP exist. So medicine directly impacted to the health. If suppose we taken the medicine and that medicines not maintain the quality as per the GMP and GLP, then definitely that will impact our body, and that can be risk of the lives also. So that's why we require the quality matters in a pharma, and to maintain the quality we need to follow the GMP and GDP. So what is mean by GMP then? GMP is nothing but good manufacturing practice. Whatever dosage form we need to manufacture in a pharmaceutical industry plant locations, that should follow the GMP practices. It is a nothing but the to maintain the quality and that consistency in a manufacturing during the every batch. That maintained should be quality and consistency. What focuses on the GMP? Clean facility should be there. Clean facility to avoid the contamination. Then a validated process should be there. Validation process, accurate, consistency, reproducibility, ruggedness, etc. etc. Then trained staff should be there. Well qualified and trained staff should be there to handling the instrument, to handle the process of the manufacturing. Then documentation also need to define after completion of the process and activity, that need to document all requirement data, and these are all regulated by the US FDA, WHO and CDSCO. WHO, these are the regulatory bodies, they maintaining the quality in different types of industries. Then what are the key principles of the GMP? There are 5 key principles available. Quality built into the product. Quality should be there. Previously side we say the meaning that quality should be there with the consistency. Then clear written procedure, that is SOP, that is standard operating procedure, should be there in a pharmaceutical industry. In a standard operating procedure, they need to mention how to operate the instrument, how to handle the process and etc. Then documentation for every steps. Documentation of the every steps, that is after completion of the process, that is a granulation, compression, coating, primary packaging, secondary packaging, that need to document in every steps and with a proper handling of the raw materials. Shifting is there, multi-mill is there, so that shipping is there, so that should be a proper handling of the raw material. The prevention of the cross contamination, to avoid this cross contamination, changeover is there, cleaning process is there, so we need to follow that. Next, what is the GLP? Good laboratory practice. The name indicates that those practices we are performing in the laboratory, that should be maintained in a good laboratory practice. Ensure quality and integrity of non-clinical data. Whatever data should be analyzed in a laboratory, that should be quality and integrity. Now, what applies to the stability studies? Different types stability studies are there. So accordingly we need to show the stability data for the expiry date and shelf life study, etc. Then toxicology reports should be generate, because any drugs or that have the toxicological impact, and that should be avoided to the patient, so we need to perform the toxicological reports as well. Then preclinical trials in vitro, in vivo study, that should be maintained. Then ensure the reproducibility and traceable data. Okay, laboratory, whatever we perform the any analysis in a laboratory, that should be reproducible and traceable during the audits. Okay, during any complications. What are the core elements of the GLP then? There 5 core elements are there. Well trained lab personnel should be there, because to performing the analysis in a laboratory, to handle the instrument, well trained personnel should be there. Okay, well educated and well trained. That personnel should be trained on SOP, trained on instrument, etc. The validated analytical method should be validated. in a laboratory to perform in the analysis. Then accurate data recording in the sense of the what hard copies and a soft copy also, okay, in computer also as well as in a hard copy also, that should be properly recorded. Then instrument calibration, in a industry there are laboratories are there, and in the laboratory there so many instruments they are used, example UV, HPLC, GC, GCMS, etc, etc. So that instrument should be calibrated at the frequency, different types of frequency available at the different industries, so according to the frequency should be calibrated that instruments. So why you need to calibrate the instrument? Because they show the proper analysis reports. If suppose we not calibrated, then that there may chances to give the false result and etc, etc. So archiving of the raw data and reports. Archiving means what? Means after completion of data, that should be archived and stored in a right place, so that we can say that archiving data, and that archiving data retrieval at the time of audit and purpose. Okay, so this is the GLP. So we seen two topics, that is the GMP and GLP in a short method, to basic concept of the GMP and GLP. So in the next video we will go towards the good documentation practice. So thank you for listening this video, and please subscribe this channel and share and like this. If you have any comments regarding the GMP and GLP, please ask me in comment box, so I will give the answer properly. Okay, thank you so much to attending the session. Thank you.